Good afternoon, everyone. My name is Dickens ABONG'O. I'm a forensic science at Grand Canyon University. Welcome to this wonderful presentation. This presentation examines how mitochondrial DNA analysis was used to identify the Romanov family. The case is one of the most influential examples of forensic genetics and demonstrates how alternative DNA testing can provide answers when traditional STR analysis is limited by degraded biological evidence. Let's look at the objectives. Today, I will explain the forensic uses of the YSTR and the mitochondrial DNA. Then I will review the Romanov case. Thereafter, I will discuss where testing was limited. Then we will explain how the mitochondrial DNA analysis solved the case. And in conclusion, we'll evaluate its significance to modern forensic science. YSTRs are inherited from father to son and thereafter trace paternal lineages. They are valuable in sexual assault investigations where male DNA is mixed with female DNA, and in missing person cases involving male relatives. However, YSTR cannot distinguish between closely related males who share the same paternal lineage. Unlike nuclear DNA, the mitochondrial DNA is inherited maternally and is present in hundreds of thousands of copies per cell. This high copy number makes mitochondrial DNA particularly useful for the degraded bones, and teeth, hair shaft, some historical remains, and mass disaster victim identification when nuclear DNA is unavailable. So on the slide we can see the different how mitochondrial DNA and the YSTR DNA is inherited from other parents. Let's discuss briefly about this case of the Romanov family. The Romanov family included Tsar Nicholas II, Empress Alexandra, and their children were executed in 1918. In 1991, skeletal remains were discovered near Yekaterinburg, that is in Russia. So scientists sought to determine whether the remains belong to the Romanov family using genetic evidence and historical records. So we could briefly see the family of the Romanov. This evidence, the evidence which was collected relied on skeletal remains, teeth, and bone samples recovered from the burial site. Researchers also used historical records and reference DNA samples from the living maternal relatives. Together, these evidence sources help establish identity and corroborate the historical account. So why traditional STR analysis was limited? And on the same slide, we could see an image of DNA sequencing. Traditional STR testing depend on nuclear DNA, which exists in only cell. After more than 70 years of burial, environmental exposure caused severe DNA degradation and fragmentation, which increased the risk of incomplete profile and allele dropout, limiting the usefulness of the STR analysis. And on this slide, we could see how DNA degeneration occurs. So scientists extracted DNA from the bones and teeth and sequenced the hypervariable regions of the mitochondrial DNA. The resulting sequence were compared with those of the living maternal relatives, and independent laboratories replicated the analysis, strengthening the confidence in the finding and reducing the likelihood of an error. And how did the mitochondrial DNA help in this case? The mitochondrial DNA analysis established maternal lineage between the remaining and the living descendants. Because mitochondrial DNA changes very slowly over generation, matching mitochondrial DNA sequences provided strong evidence that the remains belong to the member of the Romanov family despite extensive degradation. Quality assurance has been key in any scientific. Quality assurance was critical in this case. Scientists perform multiple DNA extractions, replicate testing, contamination control, peer review, and independent laboratory confirmation. These procedures ensured reliability and met forensic standards for scientific validity. What was the outcome? The investigation concluded that the remains belonged to Romanov family, and additional remains discovered later were also identified through DNA analysis. And although some controversy initially existed, repeated testing and independent verification ultimately confirmed the findings and resolved a long-standing historical mystery. So what the significance of this is that the case demonstrates the importance of mitochondrial DNA in forensic science. It establishes a model for historical identification, missing person investigations, and master victim identification. The Romanov case remains a landmark example of how alternative DNA methods can succeed when STR analysis is limited. And this work has been referenced from the reference material that attached. Thank you.